So many of you already know who Karen Kimmerle is. For those that don't, maybe don't know as much about Karen. Karen was born and raised in Oregon. She came to Hawaii in 1975. Inconceivable for me. It wasn't. A year later, she happened on an Orchid show in Kilo. That have been the Yale Orchid Society's.
Yes, it was in a completely different location.
Oh, makes sense. Well, it's probably grown, right, over the years. That kindled her interest in growing orchids. 50 years later, she and her husband Dale run Island Sun Orchids. For example, the clonal name on that Dendrobium gyrac fire twisty one on the left there is ISO because it's Island Sun Orchid, and the one who had that plant clone. See that ISO, it may very well be one of Karen's plants. They're located in Koho, at about 150 feet above sea level, the easternmost point of the Big Island. So a good climate for growing and flowering a lot of different types of orchids. That goes to cool growers. A lot of us have that problem. Some of us do not have that problem. Very jealous of you if you don't have that problem. She's also active in the Hilo Orchid Society. She's an accredited AOS judge, actually the PhD coordinator for the Hilo Judging Center. And then to quote Karen, orchids have been a big part of my life, besides providing a livelihood. I have been so fortunate to have a passion that has taught me many skills in life and allowed me to travel and meet many interesting people. And I still get excited about orchids. We were talking about that during the break. Karen will be sharing with us 1, how to decipher an orchid label, 2, explaining basic genetics, and 3, a short tutorial on researching your plants. And she'll also review with us the background of the hybrid plants from Sunset Valley Orchids that will be growing as part of our experiment, grow out experiment. And we can thank Stacy for coming up with that idea via her connection, I believe it inspired her, and introducing that as a concept for us to try. Without further ado.
Would you rather have me use this? Okay. Okay, first of all, I am not an expert. I mean, you could live 200 years and never be an expert. There's so much to learn about orchids. Is there... There's one that you could leave on so I can see what just see if that's not too distracting. Anyway, okay, so I'm not going to bore you with 100 slides. I'm going to make this as simple as possible. It's really, I was asked to make it more of a basic talk, but I hope to cover a lot of things in a short amount of time. Because I understand, I've been president of our society, I've been in the audience many times, and I know that a talk half hour to 45 minutes is the best, okay? You don't want to go really long. Okay, so the first thing is the difference between species and hybrids. Many of you know this already, but let me just go over. Species were developed in nature. Okay, that's how, and it's amazing how many varieties there are. They were developed in nature. Hybrids then are man-made. Sometimes they're made in nature too, but mostly man-made. And but the species themselves can be line-bred. So you might see some tags that have 2 species names together. We'll go over that. That's my next thing. So species and hybrids. There's also YouTubes available to go over this if you guys want to refresh yourselves about it. By the way, there's lots of resources on the internet, but YouTube's about that. So why does it matter? It doesn't really. I mean, it's a personal preference. Some people are really all about growing the species, and some people just love the hybrids, the colors, the shapes. It's a personal preference. Sometimes when you take two species that are difficult to grow and put them together, they're easier to grow. So in that way, you may rather grow a hybrid and have it be an easier grower. But for the most part, it's just preference. Again, some people, they just make it their life passion to only grow species. And then other people don't. So that's the main distinction there. So the other thing is I'm not great with PowerPoint, so this isn't any fancy PowerPoint program. So as you can see, and it's a simple 101 and then along with this hybrid exercise. So let's see here. Okay, so I hope you can see that okay. So we're going to go over tags because, well, Stacy particularly said a lot of people don't understand how a tag reads. And it seems so simple, but when I started to put it into simple terms for you, well, this is what I came up with. And once again, this is an informal talk. If you have questions, ask them. Stop me and ask them, okay? Because I wanted people to be able to understand. So you see examples of species tags. Species, the genus, that's the family that orchids are in, Vandas, Dendrobiums, Cattleyas, that's always capitalized. Species names are always not capitalized, right? Lowercase in the beginning when you write it out. So you can see all of these: Vanda curvifolium, Dendrobium moscatum, Cattleya labiata, variety rubra, that has a variety with it, and then Dendrobium tantrinum via Sib. So you're going to see all these. If you have any questions, ask me, because I'm going to do my best to explain them to you. Okay. So primary hybrids means you take two different species and put them together. So two species crossed together make a primary hybrid. Now all three of them, except the third one, haven't been named yet. That's why there's two names on them. So the first one, and then some of them have awarded parents, and that's what those letters are after, say at the top one, B. nodosa Susan Fuchs, FCC AOS, that's first class certificate. That's one of the highest awards you can get. So when you see those numbers, AM, FCC, HCC, that's an award that the plant has gotten. So look down to number 3, and that CLO stands for Cloacia, black jade. Actually, it stands for Cloacetum. Black jade, so it's Russellianum, and you can tell it's a species because it's not capitalized, by expansum, not capitalized. So that's two together. So that's a named primary hybrid. Okay?
Sharon, how does that happen? How does the naming happen?
How does the naming happen? If you make a cross or someone else has made it and it hasn't been registered, you go to the RHS website and there's a form to fill out and you can register it. It costs like $16. Last time I did it, it was $16. And wait a second now. Yeah, so that's...
So if you were to say you were going to create your own new primary hybrid, that it exists, you could actually name it as you were in the process of naming it.
Yeah, you can.
You give it its name before the seeds are even out of the class.
You can, but you ought not to. The gentleman's agreement is.
Okay.
But there are no registrations. People do it. But that's okay.
Thank you. Yes. So did everybody understand that? You should wait until it flowers first, or at least until you have plants before you name it. And you're supposed to name where the source came from, where you got the plants from. It's kind of complicated, but yes, you can. Most species have already, primary hybrids have already been made. That's not necessarily true. But yes, so you can go ahead and register. You can also register hybrids. That's not just primary hybrids, okay? So complex hybrids are ones that have 3 different kinds or more. So you can see the first one is a named complex hybrid because it has 2 parents that they're capitalized, so they're already hybrids. So it's 2 hybrids together, making that named hybrid BLC Paisley Laura Grace. The second one is named Elsie Margot because it's Jonghyuna by Luminosa. So that's Luminosa, I know, is Tenebrosa by Dowiana. So you got 3 plants in there. So it still is a complex hybrid. The fourth one, again, you see it's 2 different hybrids put together. And then the third one is a named hybrid, and it shows 2 of the parents. One is a species and one not. So again, the clue is to look whether it's been capitalized after the genus name. Okay, got that? Say again?
In the previous slide, comment on parentheses.
Oh, parentheses, okay, thank you. All tags are not created equal, okay? It's all human who put these together. So sometimes parentheses just mean that is the 2 parents. Not everybody does that. Like for instance, the top one doesn't have parentheses, but it's pretty obvious because it's written smaller and you can see the cross sign between the 2, that those are the 2 parents. But the second one and the fourth one have the parentheses, which helps you to know what the parents are, okay?
And also just add, for anybody that can't see this, Heather's recording it, so you will be able to get a copy of the PowerPoint presentation afterwards, so that you can go back and see exactly where it's from.
Okay.
And at some point, this is going to be done.
Okay, did everybody hear that? Oh, it'll be available. This PowerPoint will be available because it's being recorded. Okay, and these are clonal names. So what it means is that hybrid, RLC Volcano Peaceful, they took one of those and made a clone of it, meaning they took one of those plants, they selected out a plant and they had it cloned, meaning that they made several of it. It's a meristemming process. It's not an actual cloning process anyway. And you can tell that it's a clone because the clonal name, in this case, Volcano Queen, is in not parentheses, but what do you call it? Apostrophes. They don't use quotation marks for them, they use the apostrophes. Okay, so you see on each one, the second one, the apostrophe is missing because the person who printed it forgot to put it there. I mean, again, human. The second one, rainbow, that's the clonal name of that. There actually is one in the opportunity plants today. And the fourth one, again, Volcano Queen. Volcano Queen is the clonal name that Akatsuka orchids uses. They're the ones up in Volcano. And they grow out populations of plants, select one good one out, and then clone it, make a few thousand of those. And you can always know that that's a clone because it's in the clonal name comes after the hybrid name in those apostrophes. Okay? So I have a couple of examples here. So Cattleya tigrina, Centro de Esmeralda, variety albina. So what does that mean? It means tigrina is a species. Centro de Esmeralda is a selection of one, and it's a variety. Albina means it's an albiform. For some reason, some people use albina. I think that's the Brazilian way of saying alba. Some people use album. It's if you see alba anything, it usually means that it's the yellow or white version. Okay. And so that's crossed with Cattleya tigrina variety alba. So is this a species or is it a hybrid? Species. It's still a species because it's two tigrinas, which are species, put together, it still makes a species. Okay? It's pretty simple, really. Did you have your hand up? Yeah.
Yeah, the abbreviations for genus, are those standardized?
No, okay. I figured somebody asked me that. Somewhat, yes. If you see just to see it's Cattleya, but sometimes they get kind of obscure. And the best way to do it is to look it up on the internet. I hate to say it, but yes. And then you can, typically it will give you, I would bypass the AI because they're not always correct, but look under orchid roots or another reliable source, meaning another nursery, but you'll get the whole genus name, whether it's Cattleya, Cattleyanthe, Rhinecone stylus, whatever that genus name is. So it's just a matter of, I hate to say it, but practice. You'll understand what it means after a while. It's just too difficult to write out Cattleya for everything or Dendrobium for everything, Vanda, okay? Species or hybrid? Species, yeah. And I just put this one as an example. There's one on the table, by the way, good opportunity. I put it because sometimes certain people put in the countries it's from. Because it's a species, they just put that for extra information. Thought that was kind of cool. Oncidium twinkle, pink perfection. Species or hybrid? Hybrid, and is it a clone, right? Because it's pink perfection. Again, there's one of those on the opportunity table. There's one of those right here that's Oncidium twinkle, gold dust. There's a number of twinkles that are different clonal names. So that white and yellow one is a different clone, but they're still Oncidium twinkles. Cattleya harrisoniana, Streeter's choice by itself. Okay, species, right? Because it's not capitalized. Streeter's choice means that it was there. Okay, this was a selection out of a population of harrisonianas, and it's quite famous actually, Streeter's choice is. I think it got a high award, though that was not on the tag. But putting by itself up there means that it's not that clone anymore. It's that clone that was then bred to itself. So there's a difference. And I actually got called out on this because I had listed that plant as Harrisoniana Streeter's choice. And somebody was like, oh, wow. You know, they're thinking it's the clone. And then they get the plant and it said by itself and they were disappointed. So it makes a difference because that plant will not necessarily be the same as the clone plant. Clones, again, are virtually the same. Okay, when they clone something, it's virtually the same. There can be variation, but for the most part, they come out almost all the same. Dendrobium farmeri by Thursiflorum. And I put this one up to show that sometimes they're put in quotations, but by a sieve. So what does that mean? That means that this one plant that was farmeri by Thursiflorum, it wasn't named, was then crossed to a sibling. Sibling means that out of the population, there was, like brother, sister, but in a population of plants, a sibling was then crossed to one with each other, two siblings. And so that's what that tag means. Okay, that's kind of confusing, but okay. And one more, Cattleya clandiae, species, right? And so this is somebody who grows a lot of Cattleyas. And they had a clandia that was called bodacious bovine, okay, and then one that was called Mirtha Isabel. We probably know who this is, right? This was Ben's plant. And so he didn't bother to write cross to Cattleya clandia Mirtha Isabel, but it's understood that they're both Aclandias. Okay? That's kind of, if you look at it pretty closely, you get that? Okay.
Complete proper notation there with two clones and
Yeah, you're right. If they would have put it in parentheses, it would have been more easily understood. Correct, yeah. And then one more is Brassavola cordata. And then syn means synonymous with sublifolia. So because all these name changes, sometimes you'll see it's actually, I think, technically sublifolia now, but we all knew it as cordata. So that's what that means, is that it's synonymous with sublifolia. And then Dendrobium predator, because it's capitalized, that is a hybrid, right? And then being in the quotation, in the parentheses shows the two parents. And one more pole star, again, it's understood that Mirtha Isabel is one kind of pole star, one selection, and Sebastian Farrell was another selection. And Sebastian Farrell was awarded, by the way, HCC, put together. And true, if you had the parentheses around the Mirtha Isabel and Sebastian Farrell, then it would be more clear, but that's what that means. Okay, now I'm going to take a moment here. Because I said that I was going to talk about genetics, I'm going to talk about basic genetics, okay? Because boy, when I start to read it, I glaze over. And it is a whole study within itself. But within orchids, in my experience, basically genetics are mom and dad produce children, right? And we get genes from each one. And generally speaking, in orchids, you get 2 and 2 that make 2. And it's all, when you have 2 different parents, then you have more of a range of what you get for children. And this is very simplistic, right? But in orchids, one of the things, and this relates back to the tag too, is that it kind of matters who is the mother plant, means the one that carries the seed pod, and the father plant that gives the pollen, because the mother plant will often give more characteristics over to the progeny. So when you look at a tag, the first one listed, like mini purple cerulean there, that's the one that carried the pod. If the hybridizer is making that tag correctly, that will be the pod parent, we call it, the mother plant. The second one is the pollen parent. So oftentimes, and I'm sorry, I can't tell you which traits go with which, but the mother plant is more dominant for a lot of things, and so the hybridizer will a lot of times choose which one to hold the pollen and which one, to hold the pollen and which one to give the pollen, if they got it figured out. A lot of hybridizers, like me, I have to admit, it's like, oh, this flower is open and this flower is open. Well, let's give it a try. And a lot of times you might try it 2 different ways. Take the pollen from one, put it on the other, and likewise the pollen on that. And if you did that, sometime in your guys' experiments, you should try that and see. It takes time, right? You have to grow those plants up and see. But you'll see a difference between the progeny if one plant is the pod parent and one the pollen parent. Okay, so that's basically. And then when it comes down to color, that's a whole different thing with genetics. I did learn this the hard way. You take 2 white cattleyas, you put them together, and you get pink cattleyas. And I was like, no, can't be. So I tried it. I got pink flowers. So there's a whole genetics behind color. And I can't even begin to go into that. But it is something to consider. Just what it takes for hybridizers to get their goal for what they're trying. It takes years, by the way, too. Anyway, sometimes you get, because of the genetics, you get tetraploids. Okay, you get, let's see, ploidy is within the genes of the plant, okay? 2N is very typical. Sometimes the genes line up to give you 4N. Well, what that means is that you get, typically a plant, you'll get bigger flowers, more substance, the plant will be thicker. And you can often tell that in plants. It comes to the judging table and we'll go, wow, that flower is exceptional. It's bigger, it's darker color. You can look at the plant and see it's almost cardboard. That's typically because it has different genes, polyploidy or 4N. So again, that's a whole study within itself. But that's pretty significant too, because sometimes they take a chemical when they are flasking plants, put it in there to try and create tetraploids, because in the end, you may get more desirable traits out of it. Okay, again, that could be 3 talks in itself. Alrighty. But basically with genetics too, if you take 2 species and put them together, you're going to have a range. But if you take 2 hybrids that are complex already and put them together, you're going to get a huge range. So I did try that. I had cattleyas that I crossed together, complex cattleyas. A lot of cattleyas have a huge background, big genetic background. You put them together and I thought, oh, this is fine. I'll just cull out the good ones, save them, and then sell the rest. Well, you get a lot of junk. I mean, misshapen flowers. It doesn't always work out great. Because you have so much genetics in there, you're going to get a huge range of outcomes. Okay. And selfing, again, if you take a plant and you self it, you're limiting the genes, right? Because you're only using the genes that's in that plant. If you sieve it, meaning even if you take a sibling, you still have genes enough that you're going to get more of an outcome than if you self it. There's some purists who take the species and they self them, because I still don't quite understand this myself, but it's sort of pure, it keeps it more pure. And some people think that sieving it is a better way to go. And sometimes with species, they will not self. It's just a trait, probably, what do you call it? It's evolved in nature where they will not self with themselves. And so if you have that going and you're not getting a seed pod, that could be why. So then the good idea is to sieve it and see. So again, that's a huge talk that I'm not even beginning to qualify today, but hopefully that gives you a little bit.
Yourself, you mean that flower, its pollen and its ovum together itself, not this flower of this species, this flower of the same species.
No, correct. It's one flower, because most all orchids have the pollen and the ovary all together. And so selfing it literally means taking the pollen off of it, putting it back onto the same flower. Okay, good question though, because that can be confusing. Sieving would be to take, yeah, that's actually even saying that you have 2 siblings together. But if you take it's confusing, okay, because you could have 2 different tigrinas that aren't sieves, put them together, and you still have a hybrid.
Flower on this tree, flower on that tree.
Yeah.
Same species, they get pollinated.
They get pollinated, it's still the same species, but there's more complex genes in there because they're not the same plant. I know, okay, enough of that. Enough of that. Okay, and then I'm going to just give a little plug for AOS, because for $56 a year, you can have their online version of the magazine. I personally love the magazine, and they have almost every month they have a whole list on how to pronounce the names, which is great. I have to look at it sometimes. I don't always know. Our good friend Tom Miranda writes an article every month, the checklist. Did you guys know that? He's been doing this for 20 years, every month. I think, Tom, just use your old ones. Nobody remembers that. It's amazing to me. Yeah. So, but that's in there, all sorts of great articles. These are giveaways, so please take one if you're interested. This is another publication called Orchid Digest, full of great information, so much to learn. The AOS website.
An award in the January one, I think you have an award in there.
think one of your orchids was in there in January.
This January? I'm not sure. I don't know. Good thanks. And there's a whole section of the awarded ones from, usually it's from last year at the end of the magazine. So if you're interested in what is getting awarded, it's quite, I mean, they don't give full information, but it's great just to see really good versions of these plants. So anyway, the website itself is chock full of great stuff. I highly recommend it. If you're interested in learning about orchids, that's a place to start. But to research your orchids, well, it's good to start with the AOS, but, and they do. And they also have, along with your subscription, what's called Orchids Pro, which is a database for the awards. And you need to know the name to look it up, but it's pretty simple, and you can see, like, look up Cattleya purpurata and see how many awards it's been given. Or like, I know Matt does hybridizing, so he might want to look up and see how many awards this one has given to see if it's even worth doing. I mean, it's a very good resource. The other good resource that's on the internet is called orchidroots.com. And that was started by a couple of ladies who I happened to sit next to at this orchid meeting. There were a couple of Thai women who were I think they were engineers in their careers, and they started this database, and it's free, it's online, orchidroots.com. So if you look up, just put in Cattleya eclandii, you can see Orchid Roots, go to that, and then they'll give you a whole bunch of pictures of them. And I'll show you a little bit because I use that to put some of these up. But Orchid Roots is great. And then for species, the Internet Species, what is it called? Internet Orchid Species Encyclopedia. Super good for looking up, say, Brassavola nodosa. You go in there, you look that up, and it'll tell you what climate it's from, and it's very good database. So as much as, hey, when I first started into the judging program, which was long enough ago that we didn't use the computer, I carried the box of magazines that were our database at the time. So it's come a huge long ways. And we had this great database called Orchid Whiz, that had so much information, but the people got tired of doing it, and for some reason nobody else took it up, so that's kaput. And it was really a shame because it had all that information about the species, all that, and it would tell you all the progeny of the plant. So if you were trying to hybridize something, you could look up and see what else had been on this. Anyway, that's no longer there, but the internet's a great resource, so I suggest you do it.
As far as the goes, LCD blue stars is one of the best they will all be uniquely different, very similar, but uniquely different. And when they move, if you were to take a photograph of the roots, and you can upload it to Orchid Roots, or if you're on Facebook, you can upload it to bluemantic.com. They will take your picture of your plant and list it under this.
Yeah.
So if you're curious, when you see that tag, if you're curious of the potential of what the flowers would look like, if everybody puts in their pictures, you've got 100 pictures of LC Mini Blue Star. And LC Mini Blue Star is going to be very simple because of its genetic process. Okay. But there's some that are like bag skillets. And so it would be nice to look at it online and go, how much would be all the way to this one to this one?
Right, right. Yeah, and so that's where Orchid Roots shines, because they give you lots of examples. And so this is what I did on here. I took this from Orchid Roots. I looked up Mini Blue Star, and these are the parents. Mini Purple by Cornelia. And I know you probably can't see too well, but Mini Purple's two parents are Pumila by Walkeriana. And Cornelia is Pumila by Cattleya labiata. So Pumila is twice in there. Pumila is a very small growing plant. The flower is only about 2-3 inches wide. And so that's where the size of Mini Blue Star comes from. And you can tell, see how mini purple, not all of them are ceruleans. And everyone know what cerulean means? It means the blue form. We say blue, but it's that bluish form. See, mini purples, the one on the left is the cerulean form, but the other two are not. So in making mini blue star, you can guess that Fred used the cerulean form. So, in fact, I found the two plants that he used to make this cross that you brought in here. So, and by the way, LC Mini Blue Star is technically now Cattleya Blue Star. And I won't bore you with all the name changes, but I felt compelled to at least put up the name they're using now. I resist it. I hate that Laelia purpurata is a Cattleya purpurata now, but it's the way it is. So Cattleya Mini Purple Cerulean 4N. Again, 4N. He took a very good mini purple cerulean, had good form, good color, and crossed it to this Cornelia, which was half labiata. Labiata is your standard corsage kind of Cattleya. Okay? So that's half Pumila, small one and standard size one, crossed to mini purple. And that's actually the Floralia one that he used. It's a picture of one. Okay? And this is an example of one that Fred had awarded, SVO Blues, HCC. And this is one that I found on Orchid Roots. And it is not a named one, it's Mini Blue Star, but you can see how, again, you're looking at photos, so the color isn't always great, but you get the idea, yeah? And then another one that was just posted, the picture was posted. So when you folks flower yours, you got to take a picture and send them to Orchid Roots so they have a full database of all how they look. So, okay, let's go back though. So you have, can you expect to get out of this class? Because he used that 4N, and because the two grandparents are Pumilas, it's not going to be a big plant. Everyone's plant's going to stay compact, okay? You're almost for sure going to get that blue color because both parents are blue. And the grandparents are probably, no, I can't say that for sure. But you know, Fred, because we saw the named plants that he actually used those two, you're going to get a blue one, most likely. I mean, you know, you can never tell, some genes might jump out. But for the most part, you're going to get a nice round flower with a blue color. So it'll be interesting to see. Now, I wanted to point out that, I think it was Matt brought in, that one on the table there. It looks really similar to mini purple. Well, I had to look it up. It's called, what's it called? Bev's. Busy Bev's. And so I looked it up, and sure enough, mini purple is like the grandparent. It's in a couple of the parents, and it's much more complex than this, but it looks so very similar to this. So it's quite interesting that something so complex would look so similar. But it retained that beautiful round shape and good color. Anyway, I thought that was kind of interesting. Okay, so now we're going to go on to the Cattleya leolodoglossa. So this is an awarded plant Fred had that he then selfed. He took the pollen from that awarded plant and put it, now you can say it has a number of flowers. You can take it from one flower and put it on another flower on the same spike, right? And it's still selfed. Or you can take the pollen and put it on the same flower, same thing. That's still considered selfing because you're still using the same amount of genetic material. So this was given an FCC, and I was able to find, there's the awarded plant that he apparently used according to his tag. So it got FCC. It was a high award. It was very, look how it's the form, how the flowers hold themselves on that spike, and how they're kind of spaced out. It's a very nice flower. Anyway, so he took that and he selfed it. Okay, so what can you expect to get? Well, these are, again, from Orchid Roost. This is all different kinds of Leo lodoglossa. And by the way, it was registered by Fred Clark. He was the first one to make Leo lodoglossa, and so he registered it. And let's see. So that's just an example of the different ones. And so here are the parents. Lodoglossa is one, the seed parent, and that's Amethystoglossa by Lodogessii. And again, it come in different colors, but Fred most likely used this cerulean form. And then the parent plant was Tigrina. Now, Tigrina comes in different color forms too. And he did not say which Tigrina he used, but I'm going to make a real educated guess to say that it was the Cerulean Tigrina to get that, to get the result of that. Okay, so you self that, what are you going to get? Something similar. Hopefully you'll get the flower shape as well. Hopefully you'll get good color. You'll probably get that nice spotting. You'll get a taller plant because it's not going to change it. These are taller plants. Tigrina gets tall. Amethystoglossa gets tall. Anyway, I don't think you'll see a whole lot of variation, but grow it up and see, because I'm real curious too. I want to see everybody's photos when they've flowered these as well.
Do you have any idea what the weight is for those flowers? Do they contributors?
No, because it's not a straight species. Yeah, from that size, yeah, you could see flowers in 2 years with your guys' good sunshine and. Oh yeah, yeah.
The new growth that's on this was slightly visible when they came together, but the rest of it, the whole new growth is since November.
Since November, yeah, they'll grow fast. Yeah, so you guys, yeah, just give it good light. That's the main thing for these kind of cattleyas, is they can take really good light. So don't forget to do that. That's really important. So yeah, good luck on that one. And this is what I think Fred used, in other words. This is my guess of what he used to make that. Okay, so then the third one is, okay, Schomburgkia, Schultzii, Old Sherry. And Old Sherry, if you look that up, you don't really see it, but I've seen that plant around a lot, that clonal name, so it's just been around. And Chocolate Drop is a famous old plant. So you put those two together, and I want to stop right here and say, even Fred Clark doesn't always have names correctly. Schultzii is spelled incorrectly. I'll show you. This is how it, and it's not a Schomburgkia anymore. It's a Laelia. I know it's really a drag, but it's this is the correct way to spell Schultzii, E-I at the end. And you see the different variations. It's a very tall spike. I mean, like literally it can be a 4-foot spike. Gorgeous, gorgeous flowers. They're not always 4 feet, but they're long. That's a famous FCC, beautiful color on that. And this is Chocolate Drop. And I'm pretty sure he used this one to make this cross, okay? This is the parent of the one that you brought in. And again, it's not Cattleya, it's a Cattleyanthe, okay? So you don't have to remember that. And here, this is the plant of Chocolate Drop. So it's got this size, fully mature, right? And Schultzii, the plant is this size, but the spike is like this. You can see the spike's not long there. So on your hybrid, it's going to bring that way down. Even though the parent plant is the Schomburgkia, it's still going to shrink that spike a whole lot. Okay, so you can expect that. So Chocolate Drop itself is Guttata by Aurantiaca. Those are the two parents. And I found this one, which is very similar. This is Chocolate Drop by a different, Schomburgkia. I'm going to call it Schomburgkia. So they're somewhat similar to Schultzii is Tibacinus. So this is probably something about what you'll get the flowers to look like. But it will be interesting to see how many get a super long spike or if they stay more compact, and you'll still get a lot of flowers. And you know the color's going to be really good. Because you know Fred's had to have used a very good Schultze to make this. Old Cherry is a very good color. All right, so that's it. That's about all I wanted to go over with you. I hope you learned something. Yeah, I mean, there's so much to learn, but just I say, first of all, enjoy your flowers. Forget about whether they're hybrids. You know, enjoy them. That's the number one thing. And then just by looking and reading the tags, you'll learn them over time if you want to. That's all. So, any questions? All righty then.
Can I just add, I love using little birds. Little birds, right? So it kind of looks like yellow bird, but not quite.
What is it that you like? What is it?
You see little birds.
Oh, little birds.
So I recently learned that you just take the same names of the plants and read them, and they're still little birds. But this one might have been red, and this one might have been orange, and so they make still a little bird, but it's going to be completely different. So I have two that tag doesn't reflect it, but when Tim was here, he was like, no, I don't know it's at the nursery. One, I can't remember if it's in the Joseph Roth, I feel like it's the Joseph, Lily on Orange Grove. So one, the Joseph was the god parent. The other one, the Harmel was the god parent. One is a big, white, yellow, pink to yellow little bird. The other one is orange and red. Totally different because you switch who receives the pollen.
I love it.
Totally different plant. So this particular one, the Schultze chocolate dog one, if he did a reverse on it, could be a uniquely different plant.
It could be.
So we soon will get a list from Fred Clark, Sunset Valley Orchids. Website is a nightmare. This one doesn't have a card you can save things. You will see almost everything he has is a problem. And you will see sometimes in that this product will feel good, we reverse it. So you can get the same plant thing with parents crossing up in the other direction. So you really want to see what happens if you take this one, but turn it around and do this one. A lot of times Fred creates it.
Yeah, it's really,
So it's super cool.
it is super cool that he offers that. So you just have to have patience looking through his website because he has so many.
Return to the reciprocal cross.
Reciprocal cross.
And if you were to cross your two little birds together as their siblings, they both came from, they'd be considered siblings, they probably same two blocks. I think they're not siblings, but they belong in the same grass.
Crazy.
It gets complex, but basically they have two little birds, which is the second filial order. Filial orders being stages of inbreeding.
Did you get those from H&R? Okay.
James.
Okay, cool, yeah. Well, by the way, H&R, you know, Roy told me, I'm sure you heard him speak. So, back in the day when Cloney first started, well, I should say while it was into that, Roy was a hybridizer, that's what he did. And when he saw people taking these plants that he created and cloning like thousands of them for one of them, it really made him mad. He really hated that. So he started creating triploids. Triploids is it's even a weirder. It's 3 chromosomes to it, and what that usually does is they don't breed. They don't breed on. And he did that on purpose so that people couldn't take his plants and continue on with the breeding. And it was kind of interesting, that's all. Because at the same time, just one little thing more, at the same time, the University of Hawaii came up with breeding plants that were very similar to each other. Like it was a very big deal in the program, mostly for cut flower, where they bred these plants to where the seed pod population was all very similar. It's fascinating, really, that that was going on at the time. And anyway, the whole world of orchids has been incredible. And here in Hawaii, we've had some superstars within it. So, yeah, and Roy Tokunaga was one of them. Yeah. All right, thanks, everybody.